I want to motivate molecular dynamics simulation by way of example, and then I want to go into the detail, little bit more the physics of what these simulations are doing. And so I hope I can combine it both together. So here we have a slide on the program VMD, NMD. Damn it, how can I get rid of this thing here? I don't want to see it all the time. This is ridiculous. So, just like we did in VMD before, here we have 70,000 users, so not quite as many as in case of VMD, because NMD is a harder program to use and fewer people use it. Also, I think NMD is shared much more often because, for example, many NMD users run NMD on one of the supercomputer centers. They don't have it necessarily on their own computer. They may have it also on their own computer, but at the supercomputer center, it is then downloaded by the supercomputer center and they count only as one user. Anyway, so it's still a very popular program. It runs on small computers. I can run it on my laptop. It takes actually their advantage of the graphics processing unit. So NMD is using the GPUs, graphic processing unit, even though there's no graphics involved at all. And it's sped up by it quite a bit. And here you see that NMD is quite an innovative program from a point of view of computer technology. It used workstation clusters in 1993. You might think that's obvious, but I can tell you in 1993, all the big computer centers and main computing people used big machines at the centers. And these machines in the companies, they built them all dead today. And we realized it. And we said, we are not going to go that way. We are working rather with putting smaller computers into cluster and having them work together. And so we started this computer cluster very, very early, when it was really basically almost outlawed by the people who gave the money and so on. Then we also used in NMD a programming language, you know, probably C. You might also know C++. It is an object-oriented kind of C language. In those days, 94, everybody was using Fortran and was giving you 100 reasons why Fortran is the only good scientific computing language and C++ is a joke. And again, we realized that it's not only, you know, how fast you can compute certain things, but also a big computer program has to be engineered properly, and that we did also. So today everybody is using C++, but not in 94. In 94, I cannot tell you how many jokes people were making about us using C++. We used then commodity Linux clusters. Those were still times where companies were selling their operating system for expensive price. And we said, no, we just go with cheap Linux. It's good enough for us. And then we went to parallel computing, already with the clusters. The first parallel computer, actually in 1990, we built ourselves. So we built ourselves a 60 processor computer and showed that it's very good for molecular dynamics. And then we introduced also GPUs. Today, many people use GPUs. In 2007, we were the first one who wrote really immediately a very comprehensive paper about scientific computing with GPUs. And today, we are the leaders in petascale supercomputing. The NMD program was one of the programs that had to be tested on these new machines, called the petascale computers, before the bill was paid to Cray. So basically the manufacturers had to guarantee that NAMD was running at a certain specification, and only then was the computer paid. There were 3 programs like this, and one of them was NAMD. And we worked on this for 5 years, very, very hard, together with the vendors, to get the performance correct in the year 2013. So that was a very strong work that we had to do to reach this level. So we are really, NAMD is a very modern program, very popular, as you see, and it does also some impressive kind of calculations. Some of them I will be mentioning to you. So now if I say that VMD is a tool to think, then if I want to come up with a similar short expression for NAMD, then I would say NAMD is a tool to explore. And that's important that you have this kind of image when you're using NAMD, because you should have a simple kind of image of what you are doing scientifically. And like for example, for me, I'm a big cheese lab chief who gets computer resources that are very, very, very expensive. NAMD runs on computers that cost between $200 and $300 million. And now if you, I could have 2 attitudes. One is, oh, this is so much money. We have to be very careful. Don't do any waste, and be really precise and know exactly what you want to do. But that's not the way you do good science. The best science you do by exploring, by playing around, trying ideas. So on one side, there's this very expensive computer. On the other side, I have to tell a graduate student who basically comes through the door with hopeful eyes, say, oh, go ahead, play. I have to, of course, have a watchful eye there. But if we don't play, if we don't explore, we're not going to make discoveries. So when you are using these big ions, you still have to be in this mode of exploring, of playing around. If you don't do that, if you're too careful, you are not careful enough. If you're too careful, because you're not doing your job right. If you don't try out things and are very explorative, you are just wasting this expensive computer. So it's a difficult balancing act. Okay, so now we have this big computer runs on this big this big program runs on the big computers, and so again you think, oh, we are the fastest, we are the biggest kind of job. But if you look at our publications, you find that we had over the last, I think, during this period, 95, so 10 years, we had over 50 method papers. Papers that are basically concerned with algorithm issues. And sometimes real revolutionary algorithm. Our last revolution is the multi-level summation method that basically tries to be better than fast Fourier transformation. Fast Fourier transformation, you heard it many times. The most polished and developed algorithm you find, but it doesn't run well on parallel computers. And so we had to basically develop a solution, even though the prior solution was heavily engineered and optimized in order to do certain calculations better, the most important calculation you do on the computer, the Coulomb calculation, better. And so we had to do very revolutionary algorithm at certain times. So most of the investment in computing is actually in the algorithms and not in the straightforward computer power. If you're looking, for example, for certain computational problems in science, and how the speed up came about, then you will find out almost invariably the most speed up came from algorithmic inventions and not from computer power. And so NAMD has a lot of algorithmic inventions under the hood that make it a fast and good program. So I mentioned already molecular dynamics simulation folding. And so here we have William Headpiece folding showed a very, very brief trajectory only. And the question is then, what do you learn when you do such simulations? We fold it to the crystal structure. That's, of course, not so interesting. And what we learn is a detailed folding, how actually the protein in detail reaches the final structure. That was not known before. Before was only known before random configuration and then final crystal structure configuration. What happens in between is what the simulation shows. And here you see what I mentioned earlier. You have certain clusters that the system goes through. And here you have this timeline. And here you see that you see the secondary structure. You see at this point, 3 helices are formed. This helix, and this helix. And for at this point, the 3rd helix forms. And here with timeline, you see exactly what happens as this helix is forming. You can go there, look at it, and you can resolve it in terms of clusters. I told you I don't show the clusters here explicitly. So that is what you do. So you resolve it, but you see the detail that other methodologies cannot see. So this is the computational microscope, basically. You see, under the computational microscope, you see an entire folding event. Here we have some new folding project. It's just a pretty recent publication, where through the folding of a protein and beta amyloid fiber, that's one of the fibrils that contribute to Alzheimer, is forming. Here we have already a fibril. And here you see one in electron microscopy. You saw here then a fibril form, comes from here. This is just the end of it. And now you see how a protein adds itself to the fibril. And we can show, for example, that the fibril has, if it's a finite fibril, it has an end. There's 2 ends, but the ends are not the same. First you look at it, looks almost the same, but they're not exactly the same. One grows much faster than the other. And that we could explain for the first time. We could also explain why you have many different types of fibrils, like if you are a patient and another patient has different fibers in Alzheimer. Even in one body, you can have in the brain different fibers than in other body parts where you also have fiber growth. For example, diabetes is connected with some fiber growth too, and so on. So you see the variability that is possible. It's not just like one fiber for every patient and every part of the body. Here we see an application where molecular dynamics is particularly good, and that is in protein mechanics, as they arise, for example, in muscle proteins. In muscle proteins, we have 2 proteins, myosin and actin, that form the power stroke in the muscle, but there is a 3rd protein that's absolutely necessary, that is holding the structure of the muscle together as it contracts and expands. And so, for example, if you see a flight muscle of an insect that goes very many times, contraction, expansion, and then you see under a microscope, you think, oh, at any moment the muscle will get totally dislodged and out of shape. But it's held together by a 3rd protein. That 3rd protein is called titin. And that's a structural protein that gives muscle its elasticity and keeps it in the right geometry. And this is one of the pieces of titin. And so you can stretch titin, which is elastic, out with an atomic force microscope we have seen. And that was actually investigated by experiment and by simulation. And here you see a very interesting exemplary story about at least the old days of molecular modeling. Today, we are a little bit more fortunate and things work more easily. But in those days, for example, we had the experiment done that you just saw on the previous slide, where you stretch this protein apart, and we have the simulation. And now if you see here an axis that shows the speed with which you are tearing the protein apart, with which you pull it apart. And the speed is almost 10 orders of magnitude different between simulation, between experiment and simulation. And now, however, if you look at what is happening in the simulation, that you see on the top, you see the simulation. We simulated, and as far as we could tell, we were in close, we got a very, very interesting simulation. So look at the protein, we hold it fixed here, and we are pulling it on the ball onto the right. And now you see what happens. Here you see the experiment. The experiment shows you that you have a peak, and then the force increases and then suddenly becomes smaller until when it's totally stretched out, then you become large again, but then the protein is totally stretched. So that's trivial here, that's not interesting. Interesting is this feature here. Right at the beginning you're getting a peak. Okay, what does the simulation show? The simulation shows you stretch and it goes up, just looks like here, looks okay. And now look in this region here. And now we are stretching it out, and then we are putting it together, stretch it, put it together, stretch it. That you see what happens. You see we go over peak. And at that peak, we are breaking here in this beta strand several hydrogen bonds. These little cigarettes there are really hydrogen bonds. And so we have here, I forgot the number, I think 9 hydrogen bonds altogether that are stretched at one time because we are pulling the strands parallel. So that gives rise to that peak. Now why does it go down again? If you now pull further, then now you're getting a totally different pulling. Now you're unzipping it, one hydrogen bond at the same, one hydrogen bond at a time. So that's very, very easy to do. Whereas at this moment we had to rupture all the 9 hydrogen bonds at one moment. So we said, this is what is happening, we told them. This is what is happening. We are much faster than you guys, but I mean, we don't know this right away. We are getting this kind of feature. There's no chance that we can be wrong. Oh, you wouldn't believe what the people were saying to us. We couldn't get a single paper published, and every time the people were saying, 10 orders of magnitude, and your forces are much higher than the experiment. Yeah, they should be if you pull faster. Nobody looked at the actual picture. But then it turned out that these were the lazy thinkers. The experimentalists were not lazy thinkers. They worked very hard on the experiments, and they said, let's see if we can match up with the simulation people. Let's speed up our experiment. And then we, of course, also said, let's slow down our simulations to see that we are getting also in the same time range as the experiments. And here you see what happens. The last points here were only reached 2013 in the Nature paper. Nothing of this made ever the Nature paper. I can tell you that it hardly got to Biophysics journal. But now it was a continuum of points. You see that we are getting a slope here, and there's a theory that can describe this kind of processes in some simple manner, just a little simplification, but you can put a line through this. This comes actually from 2 theoretical biologists, Hummer and Szabo from the NIH at the time. So it looks like they lie nicely on a curve. We got it totally right. You know, when we slow it down and come together, we are getting the same force values, but the mechanism was all the time the same. But you see, sometimes you have to fight. You have to really bring things together from both sides, and very often you have to rely on experimentalists to work with you and to partner with you. And reasonable experimentalists do that. It's only the cheapies that who know what to think and so on, who go around and know everything better and know nothing. So what we discover here, yeah, we discovered here the details. You know, we really see that we are breaking right away these several hydrogens at the same time. See how it goes nicely, it's forming, and that wasn't known before. Also, what I learned that was really cool is these are these hydrogen bonds that are forming there, and my graduate student, when they did these simulations, sometimes the protein stretched out very quickly, and sometimes the protein it took forever and it didn't stretch, and I yelled at them, "What are you doing wrong? What is going on here?" And the others did it much faster. What are you doing? Until I realized I did a great injustice to the poor students. The real culprit weren't the students doing something wrong. The real culprit was water. The water molecules here, there are water molecules here, they attack the hydrogen bonds. The hydrogen bonds form between the beta strand, but once in a while comes the water and tells, "Oh, make a hydrogen bond with me, make a hydrogen bond with me." And if there are enough hydrogen bonds with the water, then they are partially broken and the rest the simulation can do. So you just have to wait for the water molecules. And so basically we didn't realize who were actually the actors on the stage, and we realized the water molecules are really the ones who are playing the role, and you know the graduate students were completely innocent. The advisor was too impatient. Okay, so you know we could do it all nicely, and that's what we learned. And now Titan has 300 of these domains, not just one. And so here you see 6 domains that were solved by crystallography by a collaborator. And now we can stretch those out. And here you see these are just 6 of 300. And now you see as you stretch them out, the system becomes straighter. And that is the main elasticity of the Titan. This breaking the bonds can happen, but only in extreme cases. Usually you stretch it just like this. So when you're stretching before you do exercise, you're supposed to stretch a little, or when you run, this is how you run it. When you are breaking one of these one of these domains, you stretch it out, you unfold it. That's what you call overstretching. Very painful, but much better than rupturing a muscle. And so what this happens is, this is normal working condition that you just saw there. Pulling one apart is rupturing the muscle. And these domains are about 30 angstrom, and when you are stretching it out, they become 600 angstrom long. So you can extend then this piece of Titan by a factor of over 20. So very good. That way you can overstretching painful, takes a few weeks, but you're getting complete healing process. So that is what we learned here. And you see also you have a theory. The average is of course very random, but you see that the shape of the curve we understand quite nicely from also theoretical description. The theoretical description is actually very elegant, and the following that, yes, as you pull this protein apart, you're doing something pretty monstrous and pretty fast. But if you would interview the degrees of freedom in the molecule, that would tell you, oh, this is nothing, very, very slow. All the other degrees of freedom, except the one that you pull, are thermally relaxed all the time. So the system is complete equilibrium, except for the degree of freedom that you are manipulating mechanically from the outside. The rest of them are in equilibrium, and so the system is always equilibrated, and that permits you to describe this process in the theory here, the curve really theoretically quite nicely. You only need undergraduates equilibrium thermodynamics to really take account of the features you see here. Here we have a some more interesting case of a also mechanical protein that you find in the gut, in our guts. You know that our gut bacteria were greatly underestimated. People say today that our intelligence, our weight, and if we have depression or not depends on our gut bacteria. And the gut bacteria, they are eating fibers, for example, and they have to attach themselves to the fiber so that they then can chemically modify them with other parts. It's the bacterium, and it basically anchors itself to the food source where the gut bacteria work on through an adhesion complex. The red and the blue proteins, they are called cohesin and dockerin. They are making a complex, and the complex is just a normal complex. You can pull it together, take it apart, put it together, take it apart. Very modular, very easy, until you pull. When you pull, it becomes as strong almost as a covalent chemical bond. So very, very strong. And so the chemical bond you can pull apart with an atomic force microscope, so we know how strong a chemical bond is, and then we know how this one is. And this one is about half of a normal chemical bond when you try to pull it apart. And so here you see a little bit how it works. So here you see, it. You see here how when you're pulling, the proteins actually, they go like this, and match better. And now we pull very strongly, and now we pull them apart. But I go back to show that we have a very sort of odd behavior. Here you see it. When you don't pull, they're loose. When you pull, they are gliding into very close fit. But then when you pull too strong, they're going apart. So basically, it's sort of like your hands go like this. They are not very tight together. Then when you pull, they move parallel. You're getting a very close fit, but then when you pull too strong, they're going apart. This way you are making what is called a catch bond, a bond that becomes stronger when you pull. And so that is what was shown here in the simulation. That is how the simulation realized, why is this bond becoming stronger when you actually pull on it? And here, this was a case of a molecular dynamics simulation winning the race against experiment. So, you know, we all want to make discoveries. So the experimentalists often win, but why shouldn't we win sometime? And so there was this protein ankyrin that was suspected to be involved in tactic sensors, for example, on the skin of fishes or in our muscles, sensors that can measure mechanical stress, for example, also in our ears, where we have vibrations. And now one of the sensory proteins that were known to be involved in sensing a mechanical strain was seen to have this protein ankyrin bound to it. And then the people got very excited, said, how does this ankyrin look like? This might be like a mechanical spring that actually can measure the tension on this receptor, and then thereby tell the body how much tension there is. And then people finally succeeded to crystallize this protein, ankyrin, and now the race was on. The experimentalists wanted to pull it, and we wanted to simulate it. And now it turns out this time we won. We published in 2005, and they published in 2006. 2 papers in Nature, of course, both Nature. Structure also pretty good, but not as good as Nature. You know, as a modeler, you always suspect it to be wrong, but particularly when there's no experiment yet. But anyway, we did it, and what we found is this is the softest spring people have ever seen. So basically, if you are having a spring of anchoring from the ceiling, and you're putting a coffee bean on it, then the spring extends by 1 meter in the gravity. So it's a very, very soft spring. And so we got the spring constant calculated, and it was within a factor 2 of the experimental one. Now this is a very, very soft spring constant. It wasn't like it was almost known. It was 10 times, 20 times smaller than people ever expected it to be. Many people thought it's actually 50 times higher. So we found very, very soft spring constant, and the experimentalists found it later. So we made the discovery and showed this data, plus a movie where you see the stretching out of the system. So here's another example. Anyway, maybe it's a question. I don't want to overwhelm you. Okay, then after this we have a question, but no, okay, so then here we have another example where actually molecular modeling is really acting like a microscope. And that is when you make nanosensors. You know that today you can make, with nanotechnology, very small sensors. The problem is only that these sensors, when they work on biological materials, have to be wet and measure biomolecules, and there are no imaging methods on that scale that tell you what is going on at the sensor, what's the sensor actually doing when it's sensing. And so the only way to actually show pictures of the sensors is to simulate the process that are going on and thereby imaging what is happening. Literally, people can get data, measurement data, but they don't know what they measure. And so here you can put everything together, and now you can do the measurement in the simulation, and then you could, and then you see what they actually measure. Now I tell you an example. This is this example that I showed you earlier. It's actually a protein that makes a channel. In this case, it's not a technical pore, but a nature pore, and through the channel goes single-stranded DNA. And so now one wants to see how that works. And so there was this king of this field, Amit Mella, and I asked Amit, can I collaborate with you? And Amit said, sure, you can collaborate, but first you have to pass a test. Oh, what test? He said that if you take DNA, single-stranded DNA, it has a 3' end and it has a 5' end, all that we know from our molecular biology textbook. And now he asked me, which end goes faster through, if you put 3' end first or 5' end first? He asked me, tell me that. If you tell me, then we can work together. Okay, so I put my postdoc to work, and so this is what we found out a few weeks later. We said it's faster when you put 3' end first. So I called Amit, we did your test. It's faster when you do 3' end first. On the phone was quiet. That's it. Klaus, that's right. But why? Tell me why. Tell me why. So he knew 3' is faster than 5', but he had no idea why. So this is exactly molecular dynamics. Molecular dynamics is seeing something, but it also sees the detail so that you can explain it. So why? What happened there? Something actually quite mysterious, that even though DNA is such a famous molecule, I thought it was known. If you are putting DNA in a small cavity like here, you find that the basis of the single-stranded DNA, this is not double-stranded DNA, just one strand of DNA, are not all like parallel, some down, some up, you know, they go basically perpendicular to the one. No, they're all in this direction, up from up to down, 3' down. They're like an umbrella like this. And now you go with an umbrella through a narrow door, it's much easier to go 3' end first through the narrow door then the opposite. And this is why 3' end goes first. The bases of the DNA are tilted all in one direction, so that they are forming a wedge, and it's much easier to go through when you are taking the sharp end of the wedge. And so that is what was shown by the simulation, and then everybody was surprised, why are these wedge lightings? But then we found out the sugar backbone that makes it very natural. It was no big deal once you look at it. Nobody knew it before, but that's how it is, okay. So that is exactly molecular dynamics simulation. You see the detail, and the detail can teach you a big lesson. Here we work today on sequencing DNA by putting it through a graphene pore. This is a graphene sheet, very thin sheet, and it's a graphene pore. And now we can count already the basis of the DNA, also in an experiment. And now we hope that we can identify the individual basis and thereby speed up sequencing. We actually have a collaboration with one of the most famous sequencing company, Oxford Nanopore, and they support some of the research actually, and so this is really in contact with real devices. So, good. Still no question. One question. I need a little rest here, and so a question excites me, gets me going again.
The DNA simulation that you had, how long did that take?
Oh, that's actually very short. It was, I think, about 10 nanoseconds or something, order 10 nanoseconds, and that would take, I would say, probably a day. We can do on many computers. We can do between 50 and 200 nanoseconds a day. Depends on computer type, and so that is just a day now. But you use a parallel computer really big, so that you have that, like you have to go max out the number of processors on the computer. You cannot have too many atoms per processor, because then actually the computer becomes slower. So here's number of processors, and let's say I have a 10,000 atom system. Then it finds out that when you go from 1 to 2 processors, to 5 processors, to 10 processors, you're getting a linear speed up. But then you go to 20, 30, then you become even, and then we go more, then you go down again. So the calculation is best when each processor has about 1,000 atoms. Why not fewer? Why not fewer? They compute fast, and that would be good, right? Every processor, 1 atom. Half an atom is not possible. 1 atom. No, because the processor is too quickly done, and then the processor wants to telephone the other processors, and the telephone lines are slow in these computers. So the processors work very fast. The telephone lines are pretty slow. You call that the communication backbone. And so you better have the processor busy, and then once in a while they pick up the phone and tell the result to the other ones, then that they are doing it too often, and then the communication backbone cannot keep up with it. So if you're doing a simulation, you can put about maybe a few fewer than 1,000 atoms, but not much fewer. And so you have then a system of certain size, and then you go that you have about 1,000, maybe 500 atoms per processor, and then you have the maximum speed, and then depends on the computer, you can go between 50, and when you are really going to the limit and you have a good computer, you can go about 200 nanoseconds. And in this case, was an order 10 nanosecond simulation, so you could have easily done in a day. So, another question?
Sure.
Okay.
You talked earlier about the multi-level summation level and the fast Fourier method.
Mm-hmm.
And that's sort of what, can you talk a little bit more about that? I didn't know.
Yeah, so what this method is doing is, it is basically, how should I put that best? It is, so basically you have all these atoms that carry charges, interact with each other. And now you have a certain way of computing it. And in the old way, where you do the fast Fourier transform, you never compute anything twice, because you think that's a waste of time. So you compute everything once, and you report all the results. And now this new method is basically saying, what the heck? The processors compute faster than the communication lines. So let's compute more and communicate less, even though we compute then double, triple. And that's basically the key point why this method is faster.
So it's
But
related
otherwise, if you read the paper, you realize why I don't tell you more. The paper has way over 100 equations. And this is the easy form. Now we are writing the big paper, which has, I think, 250 equations. Very mathematical, serious stuff. But the key idea is basically, when you want to put in the simplest word for an engineer, is that rather than communicating, let's compute it again and not bother sending it around, because that's too slow.
Okay. Can I ask a related question?
And the advantage is of this method, by the way, is the Fourier transform deals only with periodic systems. And so the old way of computing Coulomb forces was a way of assuming then, before you did the computation, that you have the same volume that you simulate infinitely periodically duplicated in the x, in the y, and in the z direction. So you really didn't compute one protein, for example, but infinitely many proteins that form a periodic lattice, because that the method required this. And now you hope that one protein behaves pretty much like all the proteins in this lattice, as long as they are far enough apart. Now the new method doesn't need to do that anymore. You can do periodic if you have it and you want it, but you can also do just a single one. You can also do a one-dimensional periodic, but not in the y and z direction periodic, and so on. So you don't have to assume periodicity anymore. And also you can calculate other forces than Coulomb. You can also, for example, calculate dispersion interactions that are the long-range interactions that fall off like 1 over r to the 6 in atoms. There, when you bring them together, you're getting a 1 over r to the 6 force energy dependence, the so-called dispersion interaction, and they can also be calculated with the method. Sorry, long answer.
No, it's good.
You had another one.
Yeah, a related question was, as someone who's sort of coming into molecular dynamics from not having been aware of this field like a few months ago, how intimidated should I be about like the fact that there are papers with 200 equations, and is there a way to sort of pick it up without and use molecular dynamics without not being ignorant of that sort of thing, but without diving completely into it and
Yeah, so I think you have several reasons to be very, very hopeful that you can manage easily. And so the reasons are several. First, on the biological side, basically it's almost impossible to study biology because there's too much. So like you cannot get a comprehensive knowledge of biology. You could say maybe the same about physics and chemistry, but to much lesser degree than about biology. Just go to the zoo, how many different animals they have in the zoo. Oh my God, it's all biology. There are too many examples. So basically, biology is something you learn on the fly, and you don't prepare for it. So that's number one. Number two, so that's more to do with what you do. Then on the other side, you know, biology has researchers from biology. They're usually not, you know, very theoretical, computational oriented. From chemistry, they are a little bit more. From physics, they are sometimes too theoretical. From mathematics, from engineering, they have a very broad range of people. As a result, they are much more communicative than the other sciences. Other sciences, they try to act like the old medical people in the Middle Ages. They talk, you know, Latin to each other to impress their patients. Probably they were talking all nonsense to each other, but all in Latin. And so pretty much today you hear the same from some scientists. Not in the life science. In life sciences, you know, they're people, they come from different fields. You don't impress them with Latin, you impress them with clear English. Yes, so you're much better, you're much more communicative, you express yourself. You have a range that you don't bother with any of the equations, you just push the button in the program, so that you understand all 100 equations if you want to. But you don't have to, you can sort of basically choose your own way. You want to just use a program which is maybe a little dangerous. I wouldn't recommend it completely that you don't know at all what's going on. You can say, oh, I really don't want to, I only want to program, I don't want to apply them. Then you learn all 100 equations, and you can go in the middle. So you have a pretty broad range there. So it's much more communicative because it's interdisciplinary, and you know, like basically the biology as a field doesn't permit you to prepare yourself anyway. You know, every system is often so different from another one that you just learn it as you investigate it. Okay, good. So you had a question?
Yeah, last is about DNA. You said it was known from the textbook that 3' goes faster than 5'.
Yeah, he knew it. He knew it. He did the experiment, Amit Mella. Nobody else knew it. So he told us to do it, and then when I told him, he got all excited because he knew 3' was faster, but he didn't know why.
But we could just look at the structures and see this alignment, like the umbrella you mentioned. What was the purpose of simulation?
The simulation showed us it go faster, and then we tried to find out why. And then we saw, when we squeezed the DNA from a very broad channel to the channel that actually was in this protein, that the side groups didn't one flap down, one flap up, but they all flapped up. So basically, you put me into the room. I have 100 arms, and now my arms are, some are down, some are like this, some are up. And now you're bringing the walls closer and closer to me, and now all my arms go up. You would have expected that, so that 50% go down, 50% go up. But all the arms go up as you squeeze me, if you squeeze me slow enough, that I'm in equilibrium with my arms. And that was not known before. Afterwards, it's easy. And those are the best results in science. The best results in science are the ones that are obvious afterwards.
Well, my question is, if they experimentally resolve the structure and look at this alignment, they might see this.
Yeah, but the alignment, nobody looked at the alignment. We are talking about real science by real people. It's not that everybody looked at everything and somewhere it's written. It's not true. So nobody knew it. Afterwards, we were surprised that we didn't know it, and nobody ever looked at it. And this is how the bases go. That is a discovery, true discovery. Okay.
You guys also able to figure out the relationship between the sizes of pore and
So that is an explanation is simple, doesn't make it bad, that makes it good. So I thought, sorry,
Is there like a point where all of a sudden, if we're able to predict what that size would be?
sorry, can you?
Is there a cutoff point where all of a sudden all of the bases flip in the same direction? Where you're able to
Yeah, the basis, it is energetically more favorable for them to be all pointing up. And so if you are now doing it slow enough, they point up. Now you say, oh, slow enough, but slow enough might be very artificial. No, no, no. You know, like for a molecule, a local relaxation, putting my arm up or putting it down, the typical time for local relaxation is about, that's already a long time, 100 picoseconds. 100 picoseconds. So that's 10 to the minus 10 seconds. Very fast. So now, whenever you do manipulation in an experiment or in a biological system, it's very, very slow compared to that relaxation. So if you are, so the systems will always have their arms up when you are bringing them into confinement that is typical for a biological or experimental situation.
It's true, but it is possible to make a transistor that you can measure on a microsecond level.
Would be, A microsecond is way too slow. Microsecond is 10 to the minus 6 seconds. That is 4 orders of magnitude slower. If you want to do an experiment to catch them, then you have to do a very fast experiment. And no atomic force microscopy can do such fast confinement at the moment. But you may try, and then you can catch some of the bases up and come down. If you do it fast enough, there should be 50-50. Yes. Okay. So now here you see how we increase the size of the simulations. And so I put in red the key numbers. So here's the years over which we sort of made NEMD do bigger and bigger systems. And also VMD, because NEMD without VMD is nothing, because you have to look at the results, and VMD is the one to look at it. And if you do large simulation, you must also have a program like VMD that can look at the large simulations. Okay, so both of these increase their sizes. In this volume, 2 nanometer cubes, you have about 10,000 atoms, like a protein lysozyme solvated in water. And here, you have an entire small organelle, namely this photosynthetic chromatophore. I think I've shown it briefly, that has 100 million atoms in this volume. Okay, so why do we want to go from here to here? Because we are interested in life. And living systems life exists in cells. A cell is the smallest living system there is. And if we want to study it, we have to understand how these community of molecules are doing their job. And now there is, of course, not a sudden transition, like everything is dead until you have the full cell. Somehow in between, these molecules begin to cooperate and do things together to make it typical for a living system. And so this one is about, for a small cell, this is about the thousandth of a volume of a small bacterial cell. So a thousandth too small for a whole living cell. But already very interesting volume, and you would probably think that this should be great to simulate. And that is where we pushed NEMD to go towards more comprehensive systems. Okay, now let's go here on the way. On the way, the first mark was here in the year 2000, where for the first time, NEMD could simulate 100,000 atoms. And that's enough to put a small membrane protein into membrane. If you have a membrane protein, membrane, water surrounding it, 100,000 atoms or more, depends on how small the system is. And so, so this was the first milestone. And so let's look what you can do with the milestone. Here you see the protein aquaporin. It's a protein that can only transport water. And the protein transports water with an amazing efficiency. It transports water very fast, but no protons. So only water without protons riding on top of them. And now you see the trick that the system is doing. When you do the simulation, you find that what happens is that you have a very narrow channel where the waters are conducting, and the water molecules come into the channel with the oxygen down in the middle of the channel at this place here. They are rotating by 180 degrees and leave with the atom up. So they go like this into the channel, rotate around and leave like this going out of the channel. And now you can convince yourself when you are doing a little, you're just back on the envelope qualitative argument that in this form, water cannot conduct protons. And so because proton conduction is a vectorial process, the protons all have one direction, but if you have two opposing directions, then it doesn't work. And so you can do that, of course, quantitatively, doing a quantum mechanical calculation, and you see you're getting a high barrier for pushing protons through water that are conducting like this. So with other words, aquaporin can conduct water molecules through the cell wall very, very quickly. The window is wide open for the water to go through the aquaporin, but no protons that normally sitting on top of water molecules can go along with it. So that is like you going through at the airport, through security, and basically this motion of water is about 1/10 of the normal diffusion of water. That means that you are coming to security like this, and then you have to slow down, and then you continue. That would be great security. And the trick that nature does is, it makes it a very narrow space. You go like this. You have to rotate, go like this. And this way they are sure you don't carry any bombs and whatever. That's very, very good trick to do a good check quickly. So it's not so easy to understand. You can read the papers. Here is a paper that we wrote on it, and there it is also explained in more detail. So, excuse me?
So you showed occasionally there's a little yellow block.
Oh, the yellow was just a tagged water, because the water is actually, in this case, the water was going randomly. It looked like it's going through one direction. In this moment, it happened to go one direction, but a few 100 nanoseconds later, it would go in the opposite direction. So we just tagged one water molecule so that you see better. So here we were. Now the next step was reached when we got about 1 million atoms, so 100,000, 1 million atoms. We could do for the first time an entire virus, a very small virus called satellite tobacco mosaic virus. And so we could simulate, and when we simulated it, it made a lot of waves. And some people got very upset with that. They say, how can you dare to simulate life? God made it, and you simulated, shame on you. But okay, so we did it anyway. And so then here we could now today simulate a ribosome, 3 million atoms. Ribosome, 300,000 atoms. If you put it in solvent with ions, 3 million atoms. Very important simulation work we are doing right now, because we have the antibiotics resistance, and many, about half of the antibiotics that are being used are actually acting on bacterial ribosomes. And so we need to know how they act there to find new candidates for ribosome, for antibiotics. And so here we study here you see the antibiotic, and it is preventing proteins, newly synthesized proteins, from going through the exit tunnel of the ribosome. And so we are not in exactly how this happened, and that will permit us to develop new ones, because we know in this particular case at least what the antibiotic is actually doing. And so, okay, so this was 3 million, and now here we have the 64 million atom system. This is the capsid of the HIV virus. It contains inside the genetic material, and this is basically the rough pattern that makes it. We have two geometries that make this capsid. One a hexagon surrounded by hexagon, and one is a pentagon surrounded by hexagon. Each of these nodes here are actually 3 proteins that are forming here a hexagonal situation. Here it forms a pentagon kind of situation. And so through electron microscopy and other and crystallography, we could actually again with people here from the University of Pittsburgh Medical School, again with Pei-John Zhang and Angela Grunenborn and other structural biologists here, we could resolve this detail, and then we could build with a computer altogether the capsid and get the full structure at atomic level. And so here you see the the simulation, 64 million atoms in a bath, and though this is how the thing looks like. And so now, what do we get out of it? We got out of it tremendous information. So here's actually the idea. It might be a little bit difficult for you to comprehend, but I can easily put it right for you very simply. What the simulation is doing here is actually much more than you think it's doing. It's actually getting together with the experimental data the structure of the system. And then we simulate with the structure and try to explain the structure and see what it's doing. But we also use the experimental data and the computer to get the structure. Why do we need a computer? Very simple. The data that go into getting the structure of this capsid are what you call multimodal. They come from different kind of experiments, from NMR structure analysis, from crystallography, from electron microscopy, and even different kinds of electron microscopies. And so to put these different information together to get a structure that is in agreement with all the different data, only the computer can do. And so here we developed a methodology for this. We call it molecular dynamics flexible fitting. So don't want to go here into further detail, but it permitted us first to get the structure together with the experimentalist, and now, once we had the structure, we could simulate it. And here we see the information we got out of the structure. First, people thought, oh, this capsid is just a container for the genetic information of the HIV virus to infect the cell, just a container. And it turned out rubbish. That is much more, it has a much more involved role. And the role is that the capsid, when it enters a cell, has to achieve to move towards the nucleus of the cell, to one of the nuclear pores, to inject through the pore its genetic material. Because the HIV virus infects non-dividing cells, where they have their genomic material all in the nucleus. So the virus has to put the genetic material into the nucleus. It can only do that with the help of the cell. And one of the key proteins of the cell is called cyclophilin, and it binds on top of this capsid in a way that is really involved real chemical detail. So we even had to use the latest atomic polarizability force field that I mentioned earlier today in order to describe this binding of cyclophilin to this capsid properly. So it's really like as complicated as when you're studying a single enzyme in detail. Here we are having a protein that interacts with this capsid, and we really needed to study it in detail, even though this protein is made of 1,300 proteins. This capsid, the blue one, 1,300 proteins. And several hundred of the cyclophilins bind with it. You think, oh, detail doesn't matter. Oh, rubbish. Detail is very, very important. And now the capsid are now being put onto the red cyclophilins are put onto the capsid in a pattern that is very regular. The reason is that the cell tries to cut the capsid open before it reaches the cell, and it uses here a protein enzyme system called E2 that tries to cut a figure 8 out of it. And now the HIV learned to put the cyclophilin in such a pattern on top of the capsid that this enzyme cannot act on it anymore. It just doesn't find its cutting paths. And so basically, the capsid uses a cell protein to help it to get to the nucleus and to prevent another cell protein from binding it. And so the interesting thing is here very simple, actually. The interesting thing is that you have a huge system made of 1,300 proteins. But detail matters. So now think of the whole cell. The whole cell is made I asked somebody how many proteins you have in your cell. 300 million. How these 300 million act with each other and form groupings, form families and villages and cities and companies and states and the whole country or this communication depends on real fine detail, on chemical detail, as much as the detail in any little enzyme that we read in the textbook about. This communication is very, very important. And so it was here that the simulation first got the structure and then got also this detail of action that, that is involved there, that is as fine as you find it in the smallest proteins just acting by themselves. Okay, so this is again another message, another lecture you learn from molecular dynamics. So I think I'm done now, and I still will begin a little bit more, so then starting tomorrow. But before I have questions, the more questions you have, the less more lecture you have to get. So then maybe you ask me a little bit. Otherwise, I shoot back and ask you questions. So, you ask me some tough questions, mean questions, like does it all make sense, or whatever. Why did you come, and do you have a dumb lingering doubt in your head?
Why did I come? To learn more about MD simulations.
And do you think they're necessarily good, or did you hear people saying, oh, don't trust them, why do you go there, you waste your time?
I've heard that.
You heard that?
Disagree with them.
So, but you disagree with them, but maybe partially they were, you worried about the arguments. Maybe we can talk about those.
The usual thing people say is you can make a computer do anything.
You can do computer. Yeah, yes and no. So my main argument there would be, sorry, tell me what have you been doing this morning? Oh, I've been doing experiment. Yeah, tell me the detail. And then your colleague will tell you, I've been sitting all morning in front of the computer. Even though he's an experimentalist. They all use computer like crazy. Why don't they apply it to their own program? Probably their program can do even more nonsense than our simulation, because we are publishing the program. You can look at it in detail. But you know, the computer basically is doing so many things today. Everybody uses it, that would be an argument I would be least impressed by. Do you agree with me, or? In the computer is used a lot. If somebody says, oh, you can do everything with the computer, can make a computer do everything, yeah, that holds probably for almost everybody. All structural biology, they use computers heavily. The beginning of structural biology coincided with a very important development in computing, namely that for the first time one could buy oneself a big computer for a department. That was the VEC 780. That was really like a serious computer. Now they could suddenly do the data analysis in their own laboratory. It was a very, very important one. And that coincided with structural biology taking off. And so you can find many examples like this. Just go through a lab, and you know, like if I go through my friends' experimental labs, I would say half of the people sitting in front of the computer, the other half have the pipettes in their hand and go like this all the time. So I don't know if you can do everything with a pipette, but you certainly can get bored with it. But, okay.
I was in a in silico drug discovery conference about a couple of months ago, and basically what people said was that investing money in nanotechnology is a good investment, for example, building iPhones, something like that. But investing money in biotechnology is a bad idea, because a lot of companies have gone bankrupt. It was a gentleman came from Merck, and they weren't happy about the progress. But they believe actually the progress, the trend is changing, and more people are becoming interested in investing, for example, in drug discovery companies. First of all, I want to see if you're, what do you think about this? Are you positive? And the second question is, okay, we are getting more powerful computers, but what about the other factors that are important in helping us, for example, designing better drugs? I've been in this field for about a year and a half only, but I don't see any significant improvement, for example, in force field parameters. we are still treating problems classical mechanically. First of all, do you think positive about this trend? Should we invest in biotechnology because of the new advancement? And also, are the computers the only factor important in making better drugs?
Yeah, for drug development,
it's a little bit bigger picture, but I think it's related to this.
For drug discovery, I have to say that I'm a physicist, and I came to biology because I wanted to understand living systems. So I'm not a medical. I'm funded by NIH actually, but I'm not a medical person. I'm really interested in basic science for human and for any other bacterium and so on. So in this regard, I stay away myself from drug discovery because that's just not my field. So that maybe not a very strong statement, but we are not doing drug discovery. So that's number one. Number two is it's like when you hear arguments how people make money and not money. It's how do these big companies make money?
I want to relate it to actually basic science because you mentioned that 15 years ago, compared 15 years ago, 10 years ago, 15 years ago to now, the molecules that we can simulate are really, really large. So we might actually be able to simulate very large molecule viruses and everything that cause these problems, and we get a better understanding just using computers how these, for example, viruses cause the problem, cause the disease, and then you'll find a better drug for them. That's what I'm talking with regard to in silico drug discovery, not other efforts that they pursue.
Yeah.
That's why I thought the question might be related.
So basically with these big molecules, we are seeing things in the context. Like for example, in case of the HIV virus, what could come out of there would possibly be a therapy against HIV. So that before we basically thought when a capsid enters a host cell, that the battle is, will the host cell cut apart the capsid before it acts? So we just thought that the capsid goes somewhere and the cell does the business, but the cell might break it apart earlier. And now that we had the full structure, we realize now that this structure is perfect for binding cell factors that actually help the capsid to go to the nucleus of the cell, whereas other cell factors, yes, try to cut it apart. But we have a communication surface that has to communicate in a positive way with the cell factor, with the host factors to go to the nucleus. So it's sort of like a different battleground that we understand it. Now, can we make tomorrow money out of it by having now a drug developed? That I cannot tell you. That's a very more complex process. And now coming back to these companies, they should not make such statements, because what are they doing? Many of them, I know if they just buy small companies where there were small people, a few people that had some brilliant idea about, they thought how a certain disease works, and they thought this chemical might interfere with it, and then they developed it, they got it through, and now then they're being bought by Eli Lilly or by some big company who doesn't have that kind of spirit and the inventiveness. They should not tell me how you discover drugs and so on. The only thing they do is buy companies. So we should rather maybe ask the small companies or the students who start them how they are doing it. But from my perspective, I would be very modest, and I would say that we try to contribute to the basic knowledge, which is closer to the living system, to the real cell of what is happening, how directly, indirectly it leads to, for example, new cancer treatments, new drugs against HIV. Sorry, I think it's a complex process. I'm really not an expert. I'm not a physical scientist. I cannot tell you that. I think personally, but that is just my gut feeling, is it's likely that as we know more and more about the cell, the information gels in some form. At some point, we know so much that now we can really develop complete strategies. We know why a drug might be, how to get the drug into the cell, why a drug may also not act properly, why we might need a drug that is patient-specific rather than having one drug for every kind of patient, and so on. So we still are accumulating a lot of knowledge, and I could imagine that that knowledge leads then quite suddenly at one point to more, to better treatments. But that is, very vague statements. I cannot make more detailed statements. So?
What can we learn from a molecular dynamics simulations? I mean, what kind of information? Because when we do molecular dynamics simulations, we are curious about the mechanism of an event. And then we make a prediction, this happens this way. And then we look at the simulation to see if that happens. But if we see that event, it only shows that this is possible. And if we don't see it, we say maybe the simulation wasn't long enough. And it's hard to say if this is how likely is this event the solution to our question. What kind of questions can we ask to
So your question is sort of a little bit, I don't mean personally or whatever, it's a little negative. Say, oh, what can we really learn from it? So then I have a counter question to you. My counter question is, so what else do we have? So basically your question is a little bit like, oh, molecular dynamics is pretty good, but it cannot do it all. Sometimes it's too slow, and then I don't see it, and so on. So it's not perfect, and so it's bad. I would rather say molecular dynamics provides you information that no other experimental method can provide you with. You see the detail, you see the dynamics. Many experimental methods don't show the dynamics. They show you static, an average picture or a static picture or a frozen picture under certain conditions that are not the biological ones. Whereas molecular dynamics can really simulate with liquids, with a proper biological surrounding. It goes into the time domain. It's very detailed. So it is in a way perfect, but it's not always perfect. But do you have anything better? Tell me what better you have.
I just want to understand better what types of questions can we ask. Should we not try to talk about how probable events are in molecular dynamics, but try to see what mechanisms are possible? Is that?
Yeah, so very commonly you ask the question when you do molecular dynamics, what is a physical mechanism or the chemical mechanism underlying a biological process? And so that is what molecular dynamics is giving you. Now that sounds a little bit formalistic, and it's actually a little bit deeper than that, because the cell, everything is going on in the cell is dynamic. And so, the mechanism that is active has to be connected with a dynamic event, and you don't see the dynamic event with experimental methodology. So you say, oh, you're just a mechanism, but it's a dynamical event that is underlying this mechanism that you can resolve with molecular dynamics, and that is pretty good. And you need it. If you don't see the dynamics, you have really a big white spot in your intellectual landscape. And then also molecular dynamics is today contributing a lot to the interpretation of experiment, in the sense that without molecular dynamics you cannot interpret the experiments. The experiments are, for example, if you do electron microscopy, you have to do a lot of computing in order to get that enter structure out of it, a dynamical process out of it. And so you basically, we are seeing more and more that molecular dynamics assist us in getting really real pictures out of experiment as opposed to just have raw data. So yeah, let's say you're doing atomic force microscopy experiment. You are seeing, you are here the time axis, and you are getting the force. You are seeing over time. What does it mean? Nothing. But if you now simulate the process and you're getting something very similar, then you say, oh, this process, the dynamic process you see in the simulation, is apparently in agreement with the experiment. So apparently this is what you see. Without the experiment, you couldn't tell they are right. But with the experiment, you say, oh, this is what the simulation really shows us. So it looks right, and now the full detail is being given by the simulation. And that is, I think, a typical example for a simulation. That it is only the simulation is providing the full information on the experiment. Mm-hmm.
You said that every time you simulate, you always see something different, right?
Mm-hmm.
They don't come up always exactly the same.
Yeah, no. I would put it differently. I would put it as follows: that every time something happens in the cell, it's different because it's stochastic. It's a stochastic system. There's a lot of stochastic factors. So when the same thing happens today, it will look clearly different tomorrow. More different, similar, different, but you always have a distribution of times it takes and so on. And molecular dynamics is doing the same.
So how many times do you repeat your simulations for you to be confident of your conclusion?
Yeah, so that is sort of a point. It's a very good point. Now today we have often error bars because we repeat simulations several times so that we can add error bars. In the old days we couldn't because the simulation was so expensive that we could only do it one time. So very often we had too few trials to have the error bars that you would expect from a normal scientific data. But that happens more today. That's sort of good about these powerful computers. You can use their power to do several parallel simulations. It's very important because you see really very often, to one's great surprise, even though we shouldn't be that surprised, that the outcomes of simulation are very distributed. And first when you see it, you say, oh my God, it's something wrong or totally chaotic. And then after a while, when you do actually enough statistics, you find you're getting a reasonable distribution. It makes really sense, and that's just the way it is. So in short, processes are random. Molecular dynamics could hardly do one simulation, so that was sort of like not a very good situation, but better to have one view than no view. And so I think it was not a mistake that molecular dynamics simulations were published, but today we can do much more simulations, and so we can sample better. For example, my group, Computer Time, I think increased over the last 2 or 3 years by almost a factor between 20 and 50 altogether. So we have much more computer time. As a result, we can, of course, do longer simulations, bigger simulations, but we can also sample much more. So that becomes much better. Mm-hmm. So maybe him and then you. So, okay.
Just to follow up on that question. So how can you be sure that your simulation is actually correct? The dynamics is actually correct. So your explanation is also correct because it's cumulative. If you get twice more, then you could lead to another and so on. So is there like special, maybe, I mean, from your experience, like one
okay,
way,
good.
So repeat the experiment in different initials, repeat it using different.
So I would say that is a very general question that you should answer yourself. Okay. What is your answer?
Well, I've read some stories that...
You make a reasonable answer, give a reasonable answer. You asked me the question, how do you know that the simulation is correct? What do you think is the answer? There's no way. How can there be a way that you're correct? Now then I ask you, how do you know that an experiment is correct? Tell me. How many ways you can tell? No way. If you do several experiments, looking at it from different points of views, then you may say, oh, there's no way it cannot be correct. I've now doing very independent experiment. So do the same in the simulation. But don't say the simulation lives in you know, territory, and there they, for some reason, everyone has to be correct. I tell you that my greatest successes as a computational scientist were simulations where I was most of the time totally wrong. So I give you an example. I had to solve the structure of a protein where the crystallographer couldn't face the crystallographic data. They only had the diffraction peaks, but no phases with it. And so they came to me and asked me, can you face that through a model, build models to see if you get the phase right. The point is now that if I get the right model and getting right phases, and I putting all the data together, I'm getting a very beautiful electron density map for the protein. Every time I have not the right model, I'm getting like really like in the sky with a lot of wind. I'm getting these very, very strange looking clouds. So first model, clouds. Second model, clouds. Third model, clouds. Clouds, clouds. Bingo! Then I had a good model, and now there is a method, it's called molecular replacement, where you iteratively can now work with the data from the experiment and with improving the model iteratively, just the way you calculate square root of 2. Once you are close enough to the point of convergence, your square root of 2 gets better. If you're too far away with the initial guess of square root of 2, you use you know, the famous algorithm, you're diverging away. So when I had the right structure, close, 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 boom. So I got it right, after being 5 times wrong. So, you know, sometimes life is simple, and you often have an all or none kind of situation, and you might hit it right. In this case, you know, I mean, knew from the convergence and from the probability of mathematics that there was no way that when we write that there could be an accident for it. But anyway, so in simulation, not a matter of how correct are you, and then you go. You have to also use your head. Very often, simulation only help you to think about the problem, and you have to modify your approach. Initially, you just get junk and junk and junk until you realize, oh my God, we forgot to put some factor in. We didn't realize it, but now that we see why things don't work, we realize it better, and so on. So this process is complicated. And same in experiments. Sometimes the only reason why you're getting a really great experiment is you submit your paper, you get it rejected, but the referee gives you some good idea because he said, no, I don't believe it if you don't do this test, you don't do this. Oh, well, let's do the test. And now you're getting, next time you're getting it into a good journal, and now it's really much firmer. So it's the same with simulation. Okay. So, yeah, so I can tell you about great sampling opportunities. So if you just want to sample a conformational space, then you do what I just described earlier here. Namely, you do replica exchange molecular dynamics, where you run your simulation at different temperatures and have transitions between them. That way, the high temperature ones go much over the landscape, and the lower ones stay more local, but since you mix them, you are getting actually, you sample a much wider territory. So this is just when you want to look at conformations. Now then you have also some other case where sampling really helps you, and that is when you want to understand a process that goes from A to B. For example, in ATP, in the synthesis of ATP from ADP by the ATP synthase. There you have a complicated motor, but you know the motor is cyclic. It goes always basically to symmetry shifted, but similar state. And then you know A and you know B. You know you go from A to go to B. By symmetry, they are related. And now now all you have to find out is what is between A and B. But you know A and you know B. And there is where sampling can help you. And that field has been developed over the last sort of 20 years, and it's called transition path sampling. And that can sample effectively transitions that are taking many milliseconds. And so because you know the beginning and the end, you are confined enough. It's much harder if you can go in any direction. That takes a long time. But if you just know I go from this corner to that corner, then you can, they have much better chances. So this is, for example, another case of sampling. And yeah, maybe in general I say that one of the hottest areas in molecular modeling today is sampling. And so you have many inventions made in regard to various situations where sampling gets to the time scale that are biologically more relevant, namely to the millisecond and beyond.
Mike, could be more precise. My question was regard to like biophysical question interaction of protein and membrane, for example. Do you suggest to change the orientation of protein to make new replication, or change the initial velocity by changing random?
So you have a protein in a lipid bilayer, and what do you want to do? You want to change the orientation of the protein or what?
No, if you want to at the end see if this interaction happened or what's the interacting domain on a protein. Then
So the protein is, let's say, a receptor that binds with
it's a peripheral protein, or you don't know exactly if this protein interacts with this membrane or not. Then you want to change it.
Oh, it's a peripheral membrane protein. You want to see if it, okay, good. Then so then you want to see if the protein interacts with the membrane. Okay, good. Yeah, so there it's actually a practical answer, because we have been dealing with lots of peripheral membrane proteins that bind to membranes, and it turns out that the ones that have really strong interaction, known interaction, that they were characterized later, they happen usually very fast. So it's not like in cases where we know this protein interacts with the membrane, now it has been found out, it has been characterized through, for example, nuclear magnetic resonance, as well as through simulations. It just takes a few 10 nanoseconds, and it establishes its contact. Sometimes, you know, how deep it goes exactly can take a little longer, but it's not like a protein goes and as the membrane, and then it hugs the membrane after an hour. No, the protein hugs the membrane right away. And then actually also one thing that really helps a lot in research when you are becoming finicky about how does it happen exactly, you have to realize that if things can go several ways, that's exactly how it goes in nature. Like, for example, people ask sometimes when you dock a protein to another protein, how can you be sure this is right? And that sounds like a really good question. It's the dumbest question you can ask. Why? Because the protein comes, interacts with this protein. In reality, one time it goes like this, one time it goes like this, one time it goes like this, one time it goes like this. If in fact nature would be so stupid that it has to go like this and then go, and then it bound, it would be totally unrobust and wouldn't work well and so on. Would be much better if you go like this, go like this, go like this. As soon as you bring the reaction partners close together, it works, you can try it out, you find it works, good enough. So people are sometimes artificially fancy, overlooking that fanciness and precision is not what is really an optimal way of working in a real cell. In a real cell, it's the opposite. It has to work under many conditions. So several of the answers that were asked here falls into the same territory, that sometimes scientists are, they have a too microscopic of a view, and they only see like the native structure, the one that has been published in some journal, but you know, often people publish only one structure where they should have published many structures. Like an NMR paper that really deals with proteins in solvent, they very often publish a whole trajectory with 50 structures. Whereas you know, sometimes they only publish one, and then you are supposed to prove that one structure, which is really nonsensical. So very often you might want to remember when you are dealing with what you call precision, that the precision you might actually be something unbiological. For example, very often students come and say, oh, professor, I have to simulate the protein, but the protein is not known in this region. What can I do? Isn't it terrible? And then we look at it, and then what you find very often is that the authors who crystallize, for example, protein, they have good arguments to say the protein is not known there because it's disordered. And then the disadvantage of the experiment becomes an advantage of the computer, because the computer is really good in describing random structures. For example, the key case is the membrane. The membrane is made of lipids that are constantly changing in the detail. We can compare nicely with NMR experiments that give us an actual tilt angle of the lipids. And we know then from that we're getting it roughly right, and we see also the different phases of the lipids reproduced in simulations. But the lipids are constantly changing. If you put a protein there, you need to know exactly at every moment where the lipids are. They are made of atoms, and you cannot just leave them out or something. You have to put them there, but they're constantly in a different form. And the same can happen within a protein that is partially disordered. You know, these things are changing around. That's the way it is, and that's the way the computer described it very well. So when you have cases where you have disorder, and you can characterize the disorder, you find that the computer are doing very well. And so then every time you're having a piece of disordered protein in a system, I say, hooray, our territory. Mm-hmm.
There's really more of an observation. It's come up several times. You've addressed it a little bit. A lot of the Well, I'm an experimentalist, so maybe I can get away with saying this. A lot of times you hear people asking, you know, molecular dynamics, how can you know? How many times do you do? Where do you know, all these things where you start from. But experimentalists, nobody asks them, you know, like, how many times, you know? A lot of times, oh, I've set it up and it's worked 3 times, right? And if they were to go to somebody with the, you know, doing an MD, they'd say, well, why didn't you start from 10 different places? And so it doesn't seem very fair. It seems a lot of these comments could be directed towards experimentalists.
Yeah, yeah. For example, that is why I like the analogy to the computational microscope, because I also like to add that is, of course, we have imaging artifacts. And you have imaging artifacts in real microscopes, but that wouldn't tell you not to use a microscope. You learn to be careful, aware of imaging artifacts, and try to recognize them and go around them, or, you know, do extra tests. But you wouldn't say, oh, this microscope and imaging artifacts, I'm not looking through it anymore. That would be completely ridiculous. You basically would say, oh, I close my eyes to data. And so the same with simulations. I think simulation can tell you a lot. You have to be, of course, careful, but they really can open your mind. And that's basically, yes, they have imaging artifacts, and you want to be aware of them. And so even if that sounds very, very fluffy to you, all of science is fluffy, you know, like you have to be aware, like you are saying in our experiment. Experiments are also, you have to be careful, and you have to use your head to avoid publishing a wrong experiment. Like when I publish papers with my young colleagues, you know, my main interest is simply, I want to be right in 20 years too. In 20 years, I want it to be still right. That's what every scientist wants, and that's what I want. So I do everything I can possibly do to be right in 20 years. But please don't ask me for a recipe. Don't ask me, how do you do that exactly? Every time it's different. But my main goal in life is to be right in 20 years. That you can, every time I send a paper, that is my only goal, not my only goal, of course, I want to announce a discovery, but you know, I want to be right, that it is, that nobody is saying all the time, oh, Schulten again wrong. Mm-hmm.
My question is about the photosynthetic systems. Molecular dynamic simulations provide accurate picture of emotions.
Mm-hmm.
You know, so, but other approaches like DFT or theoretical models predict electronic transitions and other phenomena associated with functioning of the photosynthetic system. So, is there any way to merge both approaches mathematically, or
you mean more a
do they
quantum
have to
mechanical,
be-
quantum chemical approach with the molecular dynamics approach, or what?
Yeah, like the same time are being calculated all the motions, calculate the effect of excitation.
To calculate electronic excitations. Yeah. So the first somebody asked me about, I think you, but that was a question I didn't answer you then. It's about, you know, doing force field calculation like quantum chemically for drugs. You know, we have a tool in VMD. It's called FFTK, force field. TK is the scripting language plugin, and it helps you for an unknown compound that is not described yet in the force field that you use, for example, the EMBA force field or the CHARM force field or the Gromax force field, to calculate the appropriate force field parameters. And this is an entire quantum chemical calculation you need to do, where FFTK helps you to do them. And so it's a very sophisticated program, actually for experts, and it makes the life of experts much easier than it took them in the past several months, and now we just take them several days with this program. It's called FFTK. And so there you really utilize an electronic description. Before you do the simulation, you calculate forces and then you use them in the simulation. But then you have also the situation that, for example, a chemical event happens, where, for example, two potential energy surfaces cross. You need to realize that you usually know this, and then you have to do a quantum chemical calculation connected with a classical mechanics simulation. You call that QMMM method. And I would say about a quarter of the simulations we do are QMMM calculation, because we know this is happening. And then I showed you briefly the photosynthetic system. The photosynthetic system absorbs sunlight, and it migrates an electronic excitation through the system. So you constantly have electronic down, electronic up excitation as you go through the system. This goes down, this goes up, then this goes down, this goes up, this goes down, this goes up. And so you have a lot of electronic excitation, and yes, you have to use it. And so, yes, here I talk mainly about classical molecular dynamics because it's sort of like a broader area of usage. FFTK is definitely something that also belongs to this group, because you can, if you have a compound, like in drug discovery, you have a new compound that you would try it out, you call it parameterize that process, finding the force field parameters with FFTK. And then in other cases, you have to do QMMM calculations or excited state calculations. So that's again what I call, thinking. There are many ways, and I would say almost every time a system gets really interesting, you have to do a QMMM or an excited state calculation, because these are usually the interesting points where you change the chemical nature or you do something more exotic, and that's what happens then. So you have to go beyond classical mechanics, and it's done all the time. Okay, so what you be doing? Oh, good, it's already behind. After 6, so good. Let's then end up. Thank you very much for your attention.